Please let me present about therapeutic effects of topical RCI001 on environmental and inflammation-related dry eye mouse models. Dry eye disease is the most common eye disease, and inflammation is known as major pathogenesis. Therefore, the therapeutic agents for dry eye disease are also focused on suppressing inflammation. RCI001 is a substance produced in vivo during the DNA repair process. When RCI001 is administered, it inhibits the activation of RAC1, a cell signaling molecule, and shows potent antioxidant and anti-inflammatory effects. In our previous study, 1% topical RCI001 promotes corneal epithelium healing and suppresses inflammation in the ethanol-induced ocular injury models. In another study, 1% topical RCI001 showed excellent therapeutic effects, comparable with those treated with topical 1% prednisolone, which is known to be the most powerful anti-inflammatory agent, commercially available eye drops, in the ocular alkali burn models. In this study, we evaluated the therapeutic effects of topical RCI001 on mouse models of environmental and inflammation-mediated dry eye disease. BalB-C mice induced with scopolamine patch and dry chamber and NODB10 mice were used as environmental and inflammatory DED models, respectively. Topical RCI001 was instilled twice a day for a week. Phosphate buffered saline, 1% prednisolone, and 5% lifitegrast were applied to the same models to compare therapeutic effects. Topical application of RCI001. Eye drops resulted in a significant improvement of ocular surface, staining, and tear secretion in environmental DED models compared to the PBS-treated group. Furthermore, using inflammation-mediated DED models, the topical application of RCI001 also resulted in a significant improvement of ocular surface, staining, and tear secretion compared to the PBS-treated group. In the inflammatory DED models, IL-1 beta and IL-17 levels in the ocular surface and IL-6 level in the lacrimal gland were significantly suppressed in the RCI001 group. Additionally, NOx2 and 4 in the ocular surface were also inhibited compared to PBS. Compared with other drugs, RCI001 markedly reduced the ocular staining score and promoted tear secretion, similarly with 1% prednisolone and 5% lifitegrast. Through the RAC1 inhibition by RCI001, it can suppress various inflammatory cells such as neutrophil, macrophage, and lymphocyte, and seems to be able to effectively block the complex inflammatory process in DED. In our preliminary data, long-term RCI001 administration do not induce increased intraocular pressure, which is a major side effect of the topical corticosteroid. In conclusion, topical RCI001 application showed significant therapeutic effects in environmental and inflammation-mediated DED models via stimulating tear secretion and modulating inflammation. We think that RCI001 can be a promising novel therapeutic agent in DED.